I'm Madam Joyce from Educare TZ. I'm teaching biology, and today I'll be with you to discuss our topic, that is cytology, whereby we are going to deal with our subtopic, and this is the types of cells. Whereby in the types of cells, we are going to discuss the eukaryotic cell, but we are discussing the contents which are found within the cell, and that's we can call it as organelles. So in the previous session, we terminate by looking on another organelle which is found obviously in all plant and animal cell. It is known as the lysosome. So in this session, as I promised you in the previous session, we are going to start looking on the lysosomes and the functions and the structure of it. So now you are warm welcome so that we can learn together. So what is this lysosome and its function in general? Now we can say that the lysosome, it has a lot of functions, and this one, it stands as the functioning of the lysosome, that is autophagy. We have to know what is this autophagy. The autophagy allows the orderly degradation and recycling of cellular components. So as the functioning of the lysosome, tumesema kwamba yenyewe inasaidia sana kwenye process of degradation and also recycling of cellular components. We know that within the cell we have a lot of components or organelles, so they are needed to be cycled within the cell. For example, we have microtubules, we have Golgi apparatus, we have rhizosome and so forth. So all of those organelles zinatakiwa zisambae all over the cell. So that work, it is accomplished by the presence of the lysosome in the function of autophagy. And this process occurs when cytoplasmic components become enclosed in double membrane, that is phagophore, to form a compartment known as the autophagosome for degradation. You're going to see this in the figure 1.14. So hapa tunasema kwamba hii process nzima inatokea within the cytoplasm. That's why here we have said that when cytoplasmic components become enclosed with the double membrane of the cell membrane of a cell. So pale ambapo sasa zile cellular components, or we can simply say that the cytoplasmic components, for example, let's take an example, Golgi apparatus, come closer to the cell membrane, ndipo ambapo sasa hii process ya autophagy takes place au inatokea. So simply here now you can see it form a double membrane. So the process of ule muungano sasa uliokuwa kati ya hile cellular component, any kind of cellular component, it can be either Golgi apparatus, it can be also the endoplasmic reticulum, rhizosome and so on. So it come closer to the cell membrane, ndipo ambapo inatengeneza hii kitu ambayo inaitwa phagophore, and thereby to form a compartment known as the autophagosome. So for degradation, ndipo ambapo ile process nzima ya degradation itatokea. So all of this you're going to see in the figure 1.14. So this is the regulated mechanism of a cell that disintegrates unnecessary or dysfunctional components. So hii mechanism ya autophagy inayotokea within the cell of any living organism, tunasema kwamba inasaidia sana disintegration of unnecessary or dysfunctional components. So tunajua kwamba at a time kuna muda baadhi ya organelles zinakuwa haziendelei tena kufanya kazi. So zinatokaje mule ndani, ndipo ambapo lysosome ina work upon that dysfunctional component or dysfunctional organelles which are found within the cell. So inachofanya ni kuziharibu zile components ili ziweze zikatoka nje in order for the new components to work in a proper way. But also the autophagy starts with the formation of the phagophore from the membrane. This expands and engulfs the molecule, or we can call it cargo, for degradation. So how now this autophagy inatokea? First, we have seen that there is kunakuwa na muungano kati ya ile component au ile cellular component ambayo inakuwa haifanyi kazi na cell membrane. So hiyo membrane ikishaungana pamoja na hiyo dysfunctional component, ndipo ambapo ile membrane ina expand and then it engulf the molecule or cargo for degradation. Thereby the cargo can include redundant organelles or any other chemical aggregates, and once engulfed, they are contained within the autophagosome, that is autophagy vacuole. So baada ya ile dysfunctional organelle, here simply we have seen that redundant organelle. Redundant organelle ndio hiyo dysfunctional component or dysfunctional organelle. So isipofanya kazi ndipo ambapo sasa itatolewa. First it will be engulfed. So ikibe engulfed itakuwa kwenye kama kimfuko. So that a pouch, we can call it the autophagic vacuole, ambapo ndani yake kunakuwa na ile engulfed molecule or engulfed organelle which ambayo inakuwa sasa haina function yoyote ndani ya cell. So that's the work of lysosome. But also we can say that the latter can then fuse with the lysosome, which results into release of lysosomal acid enzyme to degrade the content of the autophagosome. So baadaye sasa ndipo ambapo ile autophagic vacuole itafuse na lysosome. Ikisha fuse na lysosome ndipo ambapo ile lysosome, remember within the lysosome there is a lot of enzymes. So zile enzymes zita work upon that dysfunctional organelle ambayo sasa ipo kwenye ndani ya ile autophagic vacuole. Thereby it degrade the components or the content that are found within it, within the autophagosome. Remember the autophagosome is just like a pouch where those dysfunctional organelles they are placed. So this the content of the lysosome, that is lysosomal acidic enzyme, will degrade those dysfunctional organelles. And also we can say that the product of the degradation include amino acid among other molecules, and all of them are released back into the cytoplasm and reused in metabolism as well as in building macromolecules. So ikishafanywa sasa kazi na zile lysosomal acidic enzyme, ndipo ambapo sasa inatengeneza different kind of molecule, and one of it we have this amino acid. So ile amino acid haitatoka nje ya seli as a waste product, itaendelea kubi recycled or reused within the cell, because amino acid obviously it is used in different function within the cell. So ndio maana hapa tumesema it released back into the cytoplasm and reused in metabolism as well as building macromolecules. Remember the functions of examples of macromolecules. Here we are talking about microtubules, we are talking about interfilament and so on. All of them they are macromolecules. So zinatengenezwaje na hizi degraded components zinazotokea kwenye ile autophagosome. So that is the function of lysosome. And apart from the autophagy, but also we have another thing, that is autolysis. We have another thing, that is autolysis. So we have to also to know what the function of autolysis in lysosome. Lysosome inafanyaje kazi ya autolysis? Je, hii ya autolysis ni kitu gani? So now let's see. So autolysis is commonly known as self-digestion, which refers to destruction of a cell through actions of its own enzyme. Auto means self. So tukisema autolysis means self digestion, kwa maana kwamba seli yenyewe inajifanyia digestion, meaning that it destructs its cellular components. So it destructs the cellular components by the actions of the enzymes which are found within the same cell. So now here you can see that the lysosome breaks down to release its contents, that is the enzyme, which digests various organelles, and finally the entire cells is in certain pathological conditions. So tunasema kwamba lysosome ina release its content into a cell. Now remember, within the lysosome is whereby there is a lot of enzyme. So zile enzyme ndizo ambazo sasa zita digest all of the cellular organelles which are found within the cell. The result now here you can see finally the entire cell is in certain pathological condition, kwa maana kwamba ndani ya ile seli sasa zile organelles zote zitabe digested. The result now you can see this process is known as the autolysis or self-destruction of the cell. Why is it termed as self-destruction? Because the same cell, it releases its content, that are the enzymes. So zile enzyme ndizo ambazo zitaenda kudigest all of the cellular organelle, all of the cellular components which are found within the cell. Na ndio maana tunaitema as destruction, or we can call it self digestion, and that is the meaning of autolysis. And now you can see in our diagram, that is the lysosomal functions. The lysosomal function, we have seen that we have, that is the first, that is heterophag, we have seen already, but also we have another one, that is the the autolysis. So now you can see as this one we can call it a phagocytosis. That is the cell, it engulfs solid materials. So that process we can call it phagocytosis. So now you can see baada ya sasa ya ile cell kuengulf this solid material, inaenda kutengeneza hii food vacuole. Thereby digestion inatokea, lysosome ina release its content, ikisha release its content ina result into destruction of a cell, and that we call autophagy. Later on, now after digestion, now you can see within it also there is a place whereby after lysosome to function it properly by the function, that is the autolysis, baadaye sasa ndiyo ina release its content into the surrounding environment of the cell, and that is secretion of hydrolytic enzyme baada sasa ya kufanyiwa ile process nzima ya autolysis. But also you can see this is the normal cell. So a normal cell, remember, it contains also plasma membrane, that is a cell membrane. But also this is, as I have said, it is a normal cell because inside of it you can see there is a presence of Golgi apparatus. We have also transported vesicles ambayo ndio inaenda kutengeneza zile Golgi apparatus. Also we have rough endoplasmic reticulum. So remember, we have seen also the function of autophagy. It helps in circulation of different organelles which are found within the cell of a living organism, and that's all about the functioning of the functioning of the lysosome. So my student, now let's end here. In the next part, we're going to discuss more, or we're going to clarify more on the functioning of the lysosome. So thank you for your time and for listening.